Today will be the second lecture on the cell. Last time we studied the cell membrane, and today we are going to study what is inside the cell. So what is inside the cell is the cytoplasm, and the cytoplasm contains the cytoplasmic organelles, which are floating in the cytosol. So what is the cytosol? The cytosol is the intracellular fluid. Remember we mentioned like there is an intracellular fluid and there is extracellular fluid, which is also sometimes called interstitial fluid, which is the fluid in between the cells. And it is the cell membrane that separates between them and controls the movement of substances between intracellular fluid and extracellular fluid. So the cytosol is the intracellular fluid, and it constitutes about 55% of the cell volume, and it mainly consists of water. As you can see, 75 to 90% of it is water with dissolved components. Some of these dissolved components are large organic molecules, like the proteins, carbohydrates, and lipids. I'm saying large organic molecules because each one of them consists of smaller molecules. Like, for example, proteins are, in fact, a protein is a chain of amino acids. So smaller molecules are amino acids. Carbohydrates are groups of sugars. So the sugars are the small molecules, and so on. So it's either large organic molecules are dissolved in that cytosol, or small organic molecules, like the simple sugars, as I mentioned, amino acids, the ions, and so on. And the cytosol is the site of many important chemical reactions that take place in the cell, like those reactions that result in the formation of ATP, which is the source of energy, adenosine triphosphate, or for the synthesis of the building blocks, synthesis of protein, for example, as we will see in a moment. Also in the cytosol, there are aggregates, means large, very large molecules of non-living structures, which are called cytoplasmic inclusions. And depending on the type of the cell, there are different types of these cytoplasmic inclusions, depending on the function of the cell. They vary in their composition, they vary in their shape, and in the longevity. So, in longevity, the duration that they are present inside the cell. I will give you examples. So, for example, we have of the inclusion bodies, cytoplasmic inclusions, are the melanin granules, which are present in the cells of the skin. You can see here, this is the melanocyte, and melanocyte in the skin, we are going to study the integumentary system or the skin, maybe next week. And the melanin is produced by these melanocytes as granules, and these granules will be taken by the surrounding epithelial cells, and this melanin is responsible for the coloration of the skin. So at certain times when you are tanning, for example, there will be increased production of melanin, and there is more melanin, and then this will disappear later on. So it's not necessarily that these granules or these cytoplasmic inclusions are present throughout the entire life of the cell. Also in the liver cells, they have glycogen. Glycogen is the storage for big molecule or storage for glucose, and it's a source of energy and might be depleted in cases of famine, for example. And other cells, like the fat cells, as you can see them here, they have a lipid droplet in the cytoplasm. In fact, in a fat cell, as you can see here, that the lipid droplet occupies most of the cytoplasm, and only there is a small part of the cytoplasm in the rim, and this contains the nucleus. But most of the cell is formed of the lipid droplet. So these cytoplasmic inclusions, they vary in their composition. They could be glucose, they could be lipid, they could be pigment. They vary in their shape. They could be small, tiny granules. They could be a big inclusion, and they could be present throughout the entire life of the cell, for example, in fat cell, or they could be present at some time in the life of the cell. So far about the cytosol. As again I repeat, the cytoplasm which we are studying today consists of the cytosol and the organelles, and these organelles are the specialized structures within the cell. They are subcellular structures, and most of them are membranous. Not all of them, but most of them are membranous. So that because each one of them has a certain function, so that there will be compartmentalization of functions inside the cell. So they have membrane. Not all of them, but most of them, they have a membrane. The ones that have membrane, they could have a single membrane, and this membrane has the same structure as the cell membrane, which we have studied last time. So they have one membrane, like the vesicles. We have seen some of these vesicles last time, like the transport vesicles. And others, they have a double layer. So that, for example, in the mitochondria or the nucleus, which is also considered as a big organelle, they have a double layer. The non-membranous organelles, an example is the cytoskeleton, when we are going to study the cytoskeleton in a moment. So the organelles have different shapes, have different functions. Some of them are membranous, some of them are non-membranous. The membranous one, most of them, they have a single membrane, and the others, they have a double membrane, two membranes. The large organelle here we are going to study is the nucleus. And actually, some of the cells, they are enucleate. That's to say, they do not have a nucleus, like the red blood cells. Sometimes they are referred to as red blood corpuscles, but these red bloods doesn't mean that these red blood cells, they do not have a nucleus all their life. Actually, when these red blood cells, as we will see later on, are produced in the bone marrow, they have a nucleus. You can see here the life cycle of an RBC, red blood cell. It has a nucleus, but then before it is released to the blood, it will lose its nucleus and become enucleate. And this is important for the function of the cell, because this will increase the volume inside the cell. There's no nucleus, so there is an increase in the volume inside the cell, so you will have more hemoglobin, and then therefore you will have the cell will have more capacity to bind oxygen or carbon dioxide, so it will be more efficient in this case. Okay. On the other hand, because this cell doesn't have a nucleus, then it cannot live for a long time. It has a certain lifespan. It will be destroyed after 120 days because it doesn't have a nucleus. Not having a nucleus as well in the RBC will allow the cytoplasm, will allow the cell membrane, because it doesn't have a nucleus, to become the shape of the cell, as we will see in a moment. It is biconcave. So this will increase the surface area for the cell membrane. Increasing the surface area of the cell membrane means that there is more diffusion of gases in the cell membrane. So not having a nucleus makes the cell more efficient, but it will make it having a short lifespan. The example here is the RBC. Some other cells have more than one nucleus, so they are multinucleate cells. For example, the skeletal muscles, as we will study them later on, they have many nuclei. You can see here, like they have multiple nuclei, peripheral nuclei. They are not in the center, they are in the periphery. And the reason for that is each of these muscle cells is formed of several cells during embryonic development, and they fuse together so that we will have many of these nuclei in one cell. But most of the cells, they have a single nucleus, and this organelle plus the mitochondria is the organelle that has a double membrane. Two membranes that have the same structure of the cell membrane, and that's why it is referred to sometimes as the nuclear envelope. So sometimes we call it nuclear membrane or the nuclear envelope, because it is a double membrane. The outer part of the membrane is continuous with the endoplasmic reticulum. So here's the endoplasmic reticulum. It's another organelle which we are going to talk about in a moment. And this nuclear envelope, it has pores, similar to the ones that are present in the cell membrane. They are like gatekeepers. These pores are surrounded by integral proteins again, but they are very large pores. They are larger than the pores that are present in the cell membrane. And this nuclear membrane allows transport between the nucleus, which controls the function of the cell, and the cytoplasm. This could be either in the form of passive diffusion or active diffusion, same as we have previously studied in the cell membrane. Inside the nucleus, there are the chromosomes, and usually there are 46 chromosomes, or 23 pairs of chromosomes, because each of these 23 chromosomes comes from a parent. So in the somatic cell, there are 46 chromosomes, but in the gametes, or the sex cells, like the sperm or the ovum, there are 23 chromosomes, and when these meet together, then we will have the complete number of chromosomes, the 23 pairs, or the 46 chromosomes, one from the maternal, the others from the paternal side. Each of these molecules each of these chromosomes is, as you can see, is a long, very long coiled molecule of chromatin. This is the chromatin, as you can see. And the chromatin is formed of DNA. So this is the DNA. Purple one is the DNA. And as you can see that the DNA is in the form of a double helix molecule. So there's DNA, deoxyribonucleic acid. And then there are proteins as well. Some RNA. Together, they will form the chromatin. And the chromatin is a very long molecule here. And on these DNA and the chromatin, the genes are located. The chromatin is formed of nucleotides. We have like billions, about 3.2 billion of these nucleotides, and a group of these nucleotides will form what we call a gene. So we have like 30,000 genes in our body. Each of these genes is encoding for a certain type of protein, and each of these proteins has a certain function in the cell and in the body in general. The whole genetic material is called the genome. The whole genetic material that is present in the nucleus is called the genome, and it carries about 30,000 of these genes. The function of the genes is that they control the cellular structure, as I mentioned, because they control the protein synthesis, as we are going to talk about in a moment. So they are codes for certain types of proteins, and these proteins have different functions. The genes are also hereditary units, and they replicate during cell division, so that the new cell will have exactly the same genetic material as the original cell. Inside the nucleus, apart from the chromosomes, there is another structure which is called the nucleolus. It is called the nucleolus, and this is a spherical dark structure. Sometimes more than one nucleolus is present in a nucleus, and this is not covered by a membrane. It is present inside the nucleus, it is not covered by a membrane, and it is the site of production of ribosomes. So what are the ribosomes? These ribosomes are formed of another type of nucleic acid, which is called ribosomal RNA. So they are packages of RNA, ribosomal RNA, and proteins. As you can see, each of these ribosomes is formed of two units. There is a large subunit and a small subunit. Together, the subunits, they form the ribosome, and the ribosome chemically is formed of ribosomal RNA and protein. These ribosomes are produced in the nucleus, in the nucleolus, the part of the nucleus which is called the nucleolus, and they move through the pores of the nuclear envelope to reach the cytoplasm. They are the site where protein synthesis is done. So you can see here they are in the cytoplasm. They will attach another kind of RNA which is called messenger RNA, and on this messenger RNA, amino acids, here these are the beads which you can see in green. These are amino acids, and these amino acids will be assembled together in a chain. They have a specific sequence, so therefore they are forming a specific protein. The code for this protein is imprinted in the messenger RNA, and this messenger RNA is transcribed from the DNA. And this is how the DNA in the chromosome controls the production of proteins in the cytoplasm. Some of these ribosomes, I will talk about more details about the protein synthesis in a moment, but some of these ribosomes that are produced in the nucleolus, some of them are free ribosomes. They are just present in the cytosol, and they synthesize the proteins that are found just floating in the cytoplasm. and the others are membrane bound. They are either attached to the endoplasmic reticulum, which I'm going to talk about in a moment. So when the ribosomes are attached to the endoplasmic reticulum will become rough. So it's called the rough endoplasmic reticulum because it has attachment of ribosomes. The other side where ribosomes are attached is that they are attached to the outer layer of the nuclear envelope. So they are attached at two locations. The ribosomes are the site of protein synthesis. So what happens is that when a specific protein should be produced by a cell, this double helix molecule of DNA will split, and there will be production of what we call messenger RNA. This messenger RNA is formed of a sequence of molecules that are a template of the molecules that are present in the DNA, in that particular gene of the DNA. And this messenger RNA, therefore, it carries the codes, will move from the nucleus to the cytoplasm through the pores that are present in the nuclear envelope. When this messenger RNA reaches the cytoplasm, it will attach itself to a ribosome, because the ribosome is the site of protein synthesis. So it attaches itself to ribosome and starts collecting the amino acids from the cytoplasm. These amino acids will be transported toward the messenger RNA by another type of RNA, which is called the transfer RNA. So we have a process of what we call a translation, where what has been transcribed as a messenger RNA in the nucleus will be translated into a protein in the cytoplasm. A protein is a sequence of amino acids. Amino acids are present in the cytosol. They are carried, transferred to the messenger RNA by the transfer RNA. And so we will have like amino acid number 1, and then amino acid number 4, let's say, and then number 6, according to the code that is carried by the messenger RNA. This code is transcribed from a code that was present in the gene, in the DNA present in the nucleus, and this is how the nucleus controls the functions of the cell and the functions of the cytoplasm. Okay? And the ribosomal RNA, the other type of RNA, so now we have transfer RNA, messenger RNA, and ribosomal RNA. The ribosomal RNA is a structural RNA that is present in the ribosome, and the ribosome is the site of where protein synthesis takes place. This could be either in the rough endoplasmic reticulum, or it could be in the cytoplasm. Because the ribosomes could be free ribosomes, just present in the cytoplasm, and they will synthesize the proteins which are present in the cytoplasm, or they are membrane bound in the rough endoplasmic reticulum, and those synthesize the proteins that are needed for the cell membrane itself to replace the cell membrane, or for the proteins that are going to be secreted from the cell, to be exported, like in the form of hormones that are secreted. This is the endoplasmic reticulum, which I just mentioned. It's a reticulum means network. So it's a network of membranes, like in the form of flattened sacs that are connected to each other. And if there are ribosomes attached to them, then it is called the rough endoplasmic reticulum. And this rough endoplasmic reticulum, as you can see, is continuous with the nuclear envelope. But there is another endoplasmic reticulum which doesn't have ribosomes. So it is a smooth endoplasmic reticulum, and it is attached to the rough endoplasmic reticulum. The rough endoplasmic reticulum, because it has ribosomes attached to it, then it is a site of protein synthesis. But the smooth endoplasmic reticulum, which doesn't have ribosomes attached to it, so no protein synthesis is taking place. On the other hand, the smooth endoplasmic reticulum is used to synthesize fatty acids and steroids. So that's why, for example, you will find a lot of this smooth endoplasmic reticulum in the cells of the adrenal gland medulla. We will study that later on. They synthesize fatty acids and steroids, but the rough endoplasmic reticulum is responsible for the synthesis of protein. The other function of the smooth endoplasmic reticulum is that it detoxifies harmful substances like alcohol, some of the drugs. This is not the only site for detoxification of alcohol and drugs, but the alcohol and drugs can be detoxified in other organelles, as we will see in a moment, like the peroxisomes. The smooth endoplasmic reticulum in some other cells, like in the muscle cell, it is a reservoir. It is a site for the storage of calcium ions. Remember, these calcium ions are very important for the function of some cells, including the contraction of skeletal muscle, for example. So the calcium will be stored in the smooth endoplasmic reticulum, and the smooth endoplasmic reticulum of the skeletal muscle has a special name, which is called the sarcoplasm, or sarcoplasmic reticulum. The other organelle is a Golgi complex, named after the Golgi in the late 19th and beginning of the 20th century. Was a neuroscientist and biologist. It is formed of multiple flattened, curved, as you can see them here, membranous sacs. They look like pita bread, and they are, actually the number is variable between 3 to 20, and they are curved. So they have a convex side, which is the entry point, which faces the rough endoplasmic reticulum, and the concave side is the exit point, and that faces the cell membrane, and in between them there are cisterns, which are called medial cisterns. So these flattened sacs are called cisterns. What happens is that vesicles will move, transport vesicles will move from the rough endoplasmic reticulum, containing, for example, they contain proteins. These proteins will be processed and packaged in the Golgi apparatus, and then they will be transported out of the Golgi apparatus. So they enter on the convex side, which is facing the endoplasmic reticulum, and these vesicles, they exit through the concave side, which faces the plasma membrane. You can see them here again in this enlarged diagram. The proteins will pass from the rough endoplasmic reticulum to the Golgi complex and transport vesicles, and then it will be processed, it will be packaged. Like, for example, what I mean by processed, the protein is processed. For example, a protein is attached to lipids, so it becomes a lipoprotein. A protein will be, some of the proteins can be attached to glucose to become a glycoprotein. Like, remember the glycoproteins that were present in the cell membrane. So after the protein is manufactured, it should be processed. So the protein is manufactured in the ribosome, and most of these proteins are processed and completed and packaged in the Golgi apparatus, and now they are ready to be used. They can be either transported by a secretory vesicle. So, like, for example, the protein is a hormone, so it will be transported in a secretory vesicle. Secretory vesicle attaches itself to the cell membrane, opens, and this is what we call exocytosis. Or they can replace the membrane proteins, worn out parts of the membrane proteins, or some of these are worn out proteins, they are defective proteins, so they will be attacked and fused with lysosomes to be digested. The lysosomes are membrane vesicles that are formed in Golgi complex, but they contain digestive enzymes. And these digestive enzymes have several functions. They can digest large molecules and microbes. For example, in phagocytosis, when the bacteria, viruses, and deformed cells are phagocytized by, for example, a white blood cell or a macrophage, then it will be attacked by the lysosomes and will be digested. Or it will recycle some worn out organelles, and this is called autophagy. The organelles can replicate themselves, and the worn out ones, they will be digested by the lysosomes. Also, lysosomes are used for digestion of the entire cell, and this happens after death. This is called autolysis. After death, the tissues will be emulsified and digested by its own self, by enzymes that are present in the lysosomes, because otherwise this These enzymes are not digesting the cell because they are confined, they are compartmentalized by the membrane of the lysosome. But if this membrane leaks, then the digestive enzymes will go into the cytoplasm and digest the cell itself. Or it can be used for extracellular digestion. And this extracellular digestion, as for example, in case of fertilization. As you can see here, that the sperm has a head, and on the head of the sperm there's a very big lysosome, which is called acrosome. And when the digestive enzymes will be released from the head of the sperm, these will digest the wall of the ovum, and by this way the sperm can gain access into the ovum and release the nucleus, which contains 23 chromosomes. And so the 23 chromosomes will be attached to the 23 chromosomes of the ovum, and 46 number of chromosomes will be resumed. But here, the acrosome, which is present at the tip of the head, is digesting something outside the sperm. It is used for extracellular digestion. The other organelles which look like lysosomes are called peroxisomes and proteasomes. The proteasomes, as their name indicates, they digest protein, and the enzymes they contain are called proteases, and they digest worn out protein, defective protein. You know, it's a process of manufacturing, so some of the products are defective products. So these will be attacked and digested in proteasomes. The peroxisomes, they contain enzymes which are called oxidases, and these oxidize organic material. So like amino acids and fatty acids, they break them down by a process of oxidation. This process of oxidation results in the release of hydrogen, and hydrogen will result in the formation, excess hydrogen will result in the formation of hydrogen peroxide. Hydrogen peroxide in itself is toxic material. And so these peroxisomes also contain other types of enzymes which are called catalases that decompose the hydrogen peroxide, which is a byproduct of oxidation. So they contain oxidases that oxidize, and they contain catalases that destroys the hydrogen peroxide, the byproduct of oxidation. And again here, as I mentioned, that there are other places in the cell where toxic substances are attacked, like the alcohol and drugs are attacked. You can see here, for example, these are liver cells of a rat, normal rat, and this is a rat which has been exposed to some toxic chemical substances, and you can see that there are a lot of these peroxisomes that have been produced in the cytoplasm in order to attack the the toxic substances. The other type of organelle is the mitochondria, and the mitochondria is the type of organelle that has a double membrane. Like the nucleus, it has a double membrane. It has a central cavity, the matrix, and the inner part of the membrane has multiple folds. These are called the cristae, and these folds, they increase the surface area for metabolic for chemical reactions. So there is a lot of surface area because of the presence of multiple folds. Most of these processes, most of these chemical processes that take place in the mitochondria result in the formation of energy, ATP. So that's why these mitochondria are, in fact, they are the powerhouses of the cell. They are mainly concerned with the production of ATP. Also, the mitochondria play an important role in what we call apoptosis. Apoptosis is a programmed cell death. And the enzymes that cause this destruction are primarily produced or are primarily present in the mitochondria and released into the cytoplasm to start a chain of reaction that results in the cell death. Also, there's something else here which you should take into consideration, is that mitochondria contain part of the DNA. So not the entire DNA is present in the nucleus and the chromosomes. There is some of the DNA, very small amount of DNA, which is required for the production of proteins that are confined to the mitochondria. This DNA is present in the mitochondria. And this DNA is in fact always comes from the maternal side, because during fertilization, the DNA that comes from the paternal side, from the father's side, from the sperm, is only the nuclear protein. But the protein, the mitochondria that are present in the ovum from the mother, it is these are the mitochondria that contain the DNA, the mitochondrial DNA. So it's always from the maternal side. It is useful to follow the lineage of certain people and ancestry from their mother's side. Last point about the cytoskeleton. The cytoskeleton is the cell organelle that does not have a membrane. You remember, some of them are membranous, the organelles, others are non-membranous. The membranous have single membrane or double membrane, but this one, the cytoskeleton, has no membrane, and it's a network of protein filaments. They are either microfilaments, intermediate filaments, or microtubules, and they serve as a scaffold to support the shape of the cell. They also help for organizing chemical reactions, and they aid in the movement. Like, for example, in phagocytosis, formation of pseudopods, for example, movement of the cilia and flagella, as we will see, movement of some organelles, like the chromosomes that move from each other, away from each other during cell division. The microfilaments, as their name indicate, they are small, tiny structures. So they are very thin. They are present at the periphery of the cell, and mainly they are present in what we call the microvilli. So these are microvilli here, and they form the core of the microvillus. A microvillus or microvilli, they are a group of finger-like projections from the cell membrane. So they are finger-like projections, multiple, and so they increase the surface area of the cell membrane. So there's increase in the surface area of diffusion. That's why these microvilli are present in the absorptive cells of the intestine. For example, the small intestine. They have what we call a brush border, because there is a lot of these microvilli there. So these are the microfilaments. The others, they are the intermediate filaments. They are thicker and stronger than the microfilaments. And then we have the microtubules, which form some cylindrical structures. They contain the protein tubulin, while the microfilaments contain protein actin and myosin. And the intermediate filaments, they contain several types of protein, but the microtubules, they contain the protein tubulin, and they are assembled in what we call the centrosome, part of another organelle of the cell, which is called the centrosome, and they are important for the cell shape, movement of the cilia, and flagella. I will skip the centrosome, but just give you last few seconds on the cilia and flagella. These are, again, they are processes from the cell. Cilia are multiple and short. The flagella, where we have only one type of cell in the body that has a flagella, and that is the sperm. And these are motile. So they differ from the microvilli. The microvilli, which are for increase the surface area, they are non-motile, but the cilia, they are motile. They form sweeping movement, and the flagella will cause motility of the entire cell, like the sperm.